So this was a study where we were trying to see the effect that diabetes onset has on the change of the biochemical composition of mice. In this case, we used NOD mice, which is the classic animal model of type 1 diabetes. We were able to detect and quantify almost actually more than 700 compounds. These are small molecule biochemicals, so classic metabolites that are all in the body. So almost 60% of the 700 compounds that we quantified have changed in those mice that became diabetic. This means that the entire landscape of the metabolites of these mice changes very, very significantly with the onset of type 1 diabetes. So we have a very nice full picture of the metabolite changes that happen with type 1 diabetes onset. And the novelty here, which has not been done before, is that it's in a longitudinal method. So we follow the same mouse, and the samples were collected from the same mouse several times as they aged, and many of them became diabetic. Trying to find some metabolites that change very early on, and they might serve as indicators of diabetes onset even before diabetes actually comes up. So they would serve as early biomarkers, as we would call them, and also try to find all the changes that occur in the metabolic picture after the onset of diabetes. The next step would be obviously that try to take this and move it to patients. We have identified several early biomarkers in this animal study, so we certainly will gonna look much closer to them in patients. So we are trying to figure out a way of identifying patients and clinical trials where we can do this. So collect samples before diabetes onset and look specifically at those metabolites that we are now know are of interest. We are also thinking about using these as biomarkers, not just of diabetes, but maybe of potential treatment interventions.